Okay, so I'm really sorry about that long photosynthesis video. I didn't realize it was so long until it was done. So I can try to break that one up for you in terms of like assigning them. So you can do only the light dependent reactions and then we'll do the light dependent reactions, independent reactions or something like that. So for this one, I want to do a quick review. And then we are going to go into looking at 2 evolutionary adaptations that plants use in very, very hot weather to deal with those very hot weather. Okay, share screen. Okay. Mirror imaging and zoom. Okay. All right, so quick review. So in the chloroplasts, we have the outer membrane and we have an inner membrane. Ooh. And we also have what are called these thylakoid membranes, right? Remember, each one is a thylakoid membrane and the stacked together is a grana or granum. And then outside of that is the stroma, right? So we have light dependent reactions and the light independent reactions, right? So the light dependent and we have the light independent, right? So the light dependent reactions are going to occur in the thylakoids. And we are going to have the inputs be ADP and NADP plus, and we're going to see what comes out. We're going to have ATP and NADPH, right? But think, what else comes in? We're also going to have sun. We're also going to have water, right? And we're going to get oxygen out as well. So if we look at the light dependent reactions, we are going to see water coming in, right? Water is going to get split into 2 electrons. We're going to get a couple protons, and we are going to get oxygen, right? That oxygen is going to be coming out of the plant through the stomata, right? And remember, in the light dependent reactions, these electrons are going to be stimulated by a photon of light. They are going to move through the ETC2, right? Which is going to generate a hydrogen gradient inside the thylakoid membranes. Those H's are going to go down their concentration gradient through ATP synthase to generate ATP, right? Then we're going to have another photon of light coming in, and those electrons are going to move through ETC1, and that is going to be the final electron acceptor for that is going to be sorry, I'm not going to talk about those yet. The final electron acceptor is NADP plus, and that's going to generate NADPH, right? So here are the big outputs of the light dependent reactions. Those are going to feed directly in to the light dependent reactions, right? In the light independent reactions, this is going to happen in the stroma, right? What we're going to see for the ins is we're going to get ATP and NADPH. On the outs, we're going to kind of recycle those to make ADP and NADP plus. We're also going to see that we're going to take carbon dioxide in and we're going to get glucose out, right? So in the light independent reactions, this happens in the stroma. We're going to get carbon dioxide in. Remember that carbon dioxide is a 1 carbon molecule. It is going to come together with a 5 carbon molecule called RUBP. When those come together, you're going to get 6 carbon molecule that gets split, right, into 2 3 carbon molecules. Those are going to go along the cycle. One of those 3 carbon molecules is going to go out. The other is going to be recycled to make RUBP, right? So that's called the Calvin cycle. When you go around twice in that cycle, you can generate two 3-carbon molecules, which can come together to make C6H12O6 or glucose, which can be stored as starch, right? As an energy storage, or energy for energy storage, or you can actually go to the mitochondria and break that down through glycolysis and cellular respiration to make more ATP, right? So when you go through this, we're going to recycle ADP and NADP+, which are used directly in the light-dependent reactions, right? So that's kind of the overview to photosynthesis. We have these two parts, light-dependent and light-independent reactions. All right, so the goal of this part, however, is to talk about what happens or evolutionary adaptations for very, very, very hot climates, right? So what happens on very, very hot days is that the stomata on the underside of the leaves necessary for gas exchange that open and close need to stay closed longer because they need to prevent water loss. If the stomata are closed for too long, however, they go through something very bad, which is called photorespiration, right? Photorespiration is very bad for plants. In photorespiration, if it's very, very hot, what happens to plants is that carbon dioxide, that very, very hot, here, actually, I'm gonna do this. So in very, very hot weather, stomata stay closed to prevent water loss. And what happens is that CO2 can't get in and oxygen can't get out, right? If those stomata are closed. And that's very, very bad because plants try to use that oxygen to make glucose, but they can't because they don't have carbon there, right? And so what results is that you get a very wasteful, very unproductive process called photorespiration, where plants try to go through the Calvin cycle, but they don't have carbon to do that. All right, so plants have evolved two different mechanisms to try to prevent or circumvent this wasteful photorespiration, right? Both of these mechanisms utilize the ability to do carbon fixation into organic acids and then transfer that carbon dioxide into the Calvin cycle separately, right? And they do this in two mechanisms. The first is called C4 photosynthesis, or some plants are called C4 plants. And C4 plants, the way I remember this, they have a spatial separation of carbon fixation and the Calvin cycle. They occur in different places within a plant. And the way I remember that is C4 is a type of explosive. And so when you have an explosion, things go all over the place. So there's a spatial difference, right? An example of that are like corn plants. The other kind of plants are called CAM plants, right? And they go through what's called CAM photosynthesis. And the way I remember that is CAM has an M and so does temporal. And what temporal means is that there is a timing separation between the carbon fixation that happens and the Calvin cycle. So remember, carbon fixation in the Calvin cycle converts CO2 plus RUBP, right? To make that 6-carbon molecule, which gets split into 2 3-carbon molecules, right? And what we're gonna see here is that the carbon fixation is different in C4 and CAM plants, right? Examples of CAM plants, I forgot, jade plant and a pineapple. All right, so let's focus on C4 plants first. Right, so C4 plants reduce photorespiration in very hot weather by using a spatial separation of carbon fixation and the Calvin cycle. So let's look at what happens in C3 plants first. So C3 plants are the normal plant that we've already learned about. So in C3 plants, in normal plants, we get carbon dioxide coming in, right? When the stomata are open, so stomata are open, carbon dioxide comes in, it combines with RUBP through the enzyme that does that is Ribisco. And Ribisco is actually a really cool enzyme. It's one of the most plentiful enzymes on the planet. To make this 3-carbon molecule, we get 3 carbons coming out. We're going to get another 3 carbons to make our sugars, right? That's all happens within a normal mesophyll cell, right? In C4 plants, we're going to see that the Calvin cycle occurs in chloroplasts, not in the mesophyll, but rather in these bundle sheath cells. So here, for example, we're looking at a leaf vein. Here is where the mesophyll cells are. The bundle sheath cells are deeper in the plant, close to the vein. So in very hot climates, what we see is that some plants have evolved to do carbon fixation. Remember, taking carbon dioxide out of the air and making it into something useful, an organic molecule. Carbon fixation occurs in the mesophyll cell, right? When stomata open, and then they transfer that to a different location, which are the bundle sheath cells, right? And these bundle sheath cells then can do the Calvin cycle in a separate location, right? And this can also be a separate timing, but really the C4 plants are defined by doing the Calvin cycle in a different location than the mesophyll cells. So for example, they can use the stomata when they're open, they can do the carbon fixation, and they can transfer it to these bundle sheath cells that can do the Calvin cycle, even if the stomata are closed, right? So the CAM plants have a temporal separation or a timing separation. And in the truth of the matter, they both have a little timing differences, but the CAM plant is really all about timing. So in CAM plants, what they do is at night, when stomata are open, they do carbon fixation, carbon dioxide fixation. So they convert that carbon dioxide into some organic acid, right? You don't have to know the names of that. And then they can store that up in the nighttime. And then during the day, when stomata must be closed to prevent water loss, they can still do the Calvin cycle to create all our sugars, right? So this is just an evolutionary adaptation to allow plants to keep their stomata closed and yet still go through the Calvin cycle, right? And this all occurs just in the normal mesophyll cell. It just occurs at different times. Okay. So that's it. Remember that we have a lot of questions at the end of these PowerPoints that can be really, really helpful. All righty. Sayonara. Signing off. Bye.